Good morning, everyone. My name is Kadria Jackson. I am the Assistant Vice Chair of Quality, Safety, and Patient Experience for the Mount Sinai Health System Department of Emergency Medicine, and also the Medical Director for the Emergency Department at Mount Sinai Morningside. I will be speaking to you today on a clinical protocol developed at 2 of our sites within the Mount Sinai Health System aimed at early identification of coronavirus infection in an effort to cohort patients. I have no disclosures to report. Early in the pandemic, March to April 2020, emergency departments in New York City were overwhelmed by the number and acuity of patients presenting with acute respiratory symptoms and fever. Within the ED, we had groups of patients who were known COVID positive, known non-COVID, and patients under investigations, PUI. Given infrastructure limitations and the number of patients presenting and boarding, although a high priority, infection control was quite challenging. Best practice at that time was to cohort specific patient groups. However, there was no clinical criteria in existence to augment accurate ED cohorting. Only the COVID-19 test, which took multiple hours to come back at that time. Our objective was to study a simple COVID-19 ED screening protocol that determined the risk for a positive coronavirus PCR test result. We conducted a retrospective study of prospectively derived data from March to April 2020. The protocol was jointly developed with the Emergency Department and Infection Prevention. It was applied to patients presenting with acute respiratory symptoms, and then we evaluated the protocol against all patients presented at the time. All patients had a COVID-19 nasopharyngeal PCR done. The 5 clinical parameters utilized were cough, shortness of breath, fever, hypoxia, chest radiograph with bilateral pulmonary infiltrates. For a power analysis of 80%, we needed a sample size of 76 cases to determine the significance of difference in rates of all positive clinical variables being present between PCR positive group versus PCR negative group. These are the results obtained from 283 suspected cases of COVID-19 admitted to the hospital. As you can see, there were approximately 221 cases that were PCR positive, 22% that were PCR negative, a large percentage, 240, which had bilateral pulmonary infiltrates. When we look at the manifestation of all 5 positive clinical parameters, that was present in 51% of these cases. This is a good percentage. When you look closer based on the PCR results, we find that although, yes, individually these criteria are present in the PCR positive cohort at a high percentage, they are also present in the PCR negative cohort in a reasonable amount of cases. However, when we look at all 5 criteria, more than 50% of patients manifested all 5 clinical parameters in the PCR positive cohort, and only 4% of four patients in the PCR negative group. Interestingly enough, we were unable to determine an alternative disease etiology of those 4 cases. Several of these cases had 2 or more PCR tests. We did not have antibody testing at that time. In the end, we found that our clinical criteria had a high specificity, positive predictive value, and a 10-fold greater than not positive likelihood ratio. We conclude that our ED screening protocol during times of high COVID prevalence consisting of 5 basic clinical criteria, which were fever, cough, shortness of breath, hypoxia, and the bilateral pulmonary infiltrates, is simple to use, rapidly completed, and accurate in distinguishing PUI risk for COVID-19 infection prior to the PCR test results. Thank you. Questions?